proteins that are embedded in the membrane, right? Or sitting on the membrane. Peripheral and transmembrane or integral proteins. So we're going to talk about some different kind of proteins, and we're going to talk about how they're associated, how they actually stay kind of attached to or inside the membrane, right? And talk a bit about the transmembrane domain. All right, so that's section 1. There we go. So step 1, we're going to talk about all these proteins and how they're stuck in this membrane. But what are they doing? Why do we even care? Why do we even want proteins on a membrane? So we've got 6 functions of membrane proteins. Actually lines up fairly closely with just the 6 general functions of proteins. So kind of a nice little review of that question off the bat, right? And also a note that we're probably going to see it again on the next exam, if not the exact same question, but protein function is going to keep coming up, right? So membrane proteins can just function as transporters, right? They can form channels in a cell membrane and allow things to go in and out of the cell, right? Moving waste out, moving needed resources in and out of the cell, right? So we've got function 1. 2, they can serve, right? Proteins, enzymes are proteins. So if you have reactions that need to go on around or near a cell membrane, right? You've got proteins that function as enzymes, catalyzing reactions from one thing to another. They also serve as cell surface receptors, right? We talked about kind of that extracellular matrix that is sensing things outside the cell, and it tends to be attached to the cytoskeleton inside, right, to transmit messages, and those messages go through these membrane proteins, right? Things that are happening outside of the cell, maybe some kind of chemical signal, right, gets transmitted through these membrane proteins to the inside of the cell, right? So these cell surface receptors, right, like message receptors. Another one that we've mentioned are these cell surface identity markers, right? These proteins that have phospholipids or glycolipids as little tags, right? That are kind of serving as those little cell name tags to kind of let other cells know what kind of cell it is and what organism it belongs to. These identity markers. We talked about cell junctions, right? So cell to cell adhesion is also something done by membrane proteins, things like proteins like adhesins that act in adhesion junctions, right? Where you have these 2 proteins that are attaching these 2 cells together, right? And then again, kind of interconnected with some of these other functions, you have proteins that are attached to cytoskeleton fibers, right? And these allow cells to have specific structures, right? Again, it can be involved in messages going in and out of the cell, but like this sort of structure where you've got a protein in the membrane hanging onto fibers could be the kind of thing that's set up in a red blood cell, right? Where you have 2 of those proteins taking what would normally be kind of a globby cell and like kind of winching them together, right? So you get that kind of flattened red blood cell shape, right? That is done by membrane proteins that are attached to the cytoskeleton inside the cell, right? That provide that structure. Right, so we've got these 6 functions of membrane proteins. Given the diversity of function, they can have kind of a diversity of structure, a different, kind of several different ways that they can interact with and be attached to that membrane. Right? And so let's talk about how they get attached to the membrane. So we can have peripheral proteins. If you remember when we were talking about kind of the fluid mosaic model, right? You've got this kind of fluid lipid membrane, you've got proteins floating on it and proteins that are kind of stuck in it. So we'll start with the proteins floating on it, the peripheral proteins, right? And so these are anchored to the membrane, usually with some kind of extension of these protein molecules, right? So you may have some protein out here, right, that's doing something enzymatic or something, and then it's going to have some kind of long tail that's going to anchor it to the lipid membrane, right? And so the thing is to interact with the bilayer, right? The problem is, right, it's got these nice polar heads, but the inside of it is non-polar. So you need something non-polar to anchor you into a lipid bilayer. So they'll frequently have these non-polar regions that will kind of by hydrophobic exclusion get pushed into the membrane and serve as an anchor. Right? And then you can have kind of the functional domain right, of the protein kind of floating up above. Right? And so when we talk about regions of proteins, right, we've got this kind of anchoring domain, this kind of functional domain, and then a chemical bonding domain, right, that attaches the two so that this protein stays stuck to its little anchor in the membrane. So we can have proteins attached to the outside. Right. We also have integral proteins, right? So the proteins stuck on the outside of, they're kind of hanging out outside the membrane, but you've also got proteins embedded in, like going all the way across membranes, like protein bridges through a membrane. And so these are held in place by non-polar domains, right? Lipid bilayers are polar on the outside and then non-polar on the inside. So you can't put polar stuff in here. It's going to get pushed out, right? Non-polar stuff is going to get pushed in. So if you have a protein, right, that folds up into its final tertiary structure, and it has kind of this non-polar middle, right? It's going to get pushed down in with all the other non-polar stuff in the membrane. Right? So you've got these kind of polar outsides, kind of hanging out with the polar heads of the lipids, and then this non-polar middle that gets pushed into the membrane, and it's stuck there. Once it's here, it stays here. Why? What's happening? What behavior of water is going to keep those proteins stuck in the membrane like that? Not dehydration, right? So what does water do to non-polar things? It pushes them away, right? What do we call that? That when water pushes away non-polar or hydrophobic things?
Hydrophobic exclusion.
Hydrophobic exclusion, exactly. Right? Because that's what you've got this kind of, right? We're all just kind of aqueous environments. And so the water is going to push this non-polar stuff into the same place that it's pushed every other non-polar thing it's got, right? It all just kind of gets clumped together. And so once these non-polar regions are in this non-polar kind of area, this non-polar domain of the protein is stuck in the membrane, right? So we've got these integral proteins that get anchored inside the membrane. All right, by hydrophobic exclusion. All right, so these transmembrane proteins can take a few different forms. We'll get too kind of out into the weeds here, but we'll talk about them briefly, right? You can have, so when we talked about secondary structure in proteins, right, they can form those coils, those alpha helices, or they can kind of form these wide flat pieces that we call beta sheets. All right, now the helices can do a couple of things, right? You can have a protein that's just got this one little kind of helix piece that has one of those non-polar domains, right, that would serve as an anchor, right? We call this a single pass because it just goes into the membrane once, right? It just passes through the membrane one time. You can have a more complicated protein with lots of these kind of non-polar domains. And so that protein is going to fold up, right, and pass back and forth through the membrane. Right? If the membrane is right here, right, it's just going to pass back and forth, and that's a multi-pass membrane, right? So these, you see these more with like kind of enzymatic, these are used like frequently enzymes or transporters or things like that. With this single pass, this is, and it kind of has that look to it, right? You have a single pass and then you have some tag on the outside, right? These are things like cell recognition or surface receptors, things like that, right? They're just anchored here with some piece of a protein doing something outside the cell. Yeah. If we have a protein that has a whole lot of those flat beta pleated sheets, and that transmembrane protein kind of folds itself up, what frequently happens, right, in this situation, so it's these flat beta sheets, and the flattened sheets will be polar on one side and non-polar on the other, like a piece of paper that's a different color on each side, right? The non-polar sides are going to come into the membrane and face the lipids. So as this thing kind of folds up, it ends up kind of forming a tube through the membrane where the non-polar sides all face out and the polar sides face in to this kind of tunnel that it makes. So these are channel proteins or transporters, right? Because they make this kind of polar channel or polar tunnel through the membrane, right? And this is going to be how we get stuff in and out of the cell, is through these kind of polar tunnels that get made of these beta-pleated sheets. Right. So we've got a few different ways that these transmembrane or these integral proteins can kind of form. All right, so as I kind of alluded to, a lot of these transmembrane or integral proteins are involved in transporting things in and out of the cell, right? Getting through that nonpolar lipid bilayer, right? So we're going to talk about passive transport, which doesn't use energy. So we're going to talk about diffusion. We'll talk about a couple proteins that do diffusion, right? And we may or may not get into osmosis, depending on what time is like by the time we get there. So passive transport is just when things are transported in and out of a cell and no energy is used. Okay? This is generally due, at least in part, to a concentration gradient. You're going to get this in lab, but unfortunately, I think not until after our exam, if I have my dates vaguely correct. But what a concentration gradient is, right? A gradient is just, right, like a grade, right, like a slope. So on a concentration gradient, you have kind of an area where there's a whole lot of something, and then an area where there's not a lot of something, right? If you can see this if you do things like milk in coffee or tea. If you have like Neo, you like drop in water and you don't stir it, right? You can see where you've put a high concentration of something into a liquid and it diffuses, right? It kind of spreads through the liquid over time, right? If you drop dye or something in water and you just stare at it for a while, right? The drop is initially very highly concentrated, but molecules are kind of always moving just a little bit. And so just naturally, right, by just a random kind of net movement of molecules in something, right, whatever you're putting into that liquid will kind of diffuse around. They'll go from wherever they're highly concentrated to a low concentration until everything's just kind of evenly distributed. Right, so this is called moving down a concentration gradient. So things tend to move from where there's a high concentration to where there's a low concentration due to this just kind of random movement of molecules. So you can have this kind of natural diffusion that takes place in liquids, right? Called diffusion. But we've got this problem again of this hydrophobic cell membrane interior. So things would just kind of diffuse in and out of the cell from wherever there's a lot of them, wherever there's a little of them. But we've got this hydrophobic layer. And so no, especially larger polar molecules aren't going to be able to just kind of wiggle through that, right? They're not going to be able to get through that non-polar barrier, right? Non-polar things can, right? Non-polar things can wiggle through a non-polar membrane. So things like oxygen, right? That your red blood cells are dropping off at tissues, that O2 can be released and it will just diffuse into cells naturally. The barrier, the lipid layer is not a barrier. Same thing like hormones, steroids and things, those will just naturally diffuse. But a lot of the things your body needs are polar, or at least charged, right? So how did those get across the membrane? Because they need to get into the cell somehow. And so we can't just use regular diffusion, right? Like we would for oxygen steroids. We have what's called facilitated diffusion. So like diffusion with a little bit of help, right? So we still have molecules going from like down their concentration gradient, right? Where there's a high concentration, where there's a low concentration. But they go through these membrane proteins that we've been talking about, right? They take a couple different forms, right? You can have a channel protein or you can have a carrier protein. Right, so channel proteins, we specifically mentioned, let's talk about those for just a tick. So a channel protein, right? Like those rings of beta sheets that are hydrophobic on the outside and hydrophilic on the inside. Right, so you can have a protein, and we'll just draw them like as tubes now because it's like easier, right? That has a hydrophilic interior, which means that polar things or ions can pass through them, right? You've made a barrier between yourself and this hydrophobic layer, right? So we can have these ion channels where these different kind of charged ions can pass through this channel protein, right, from outside to inside the cell, or vice versa. Right? And so how fast these atoms or molecules move depends on a few different things. One is the concentration, right? Things are going to diffuse from wherever there's a lot of them. So in this case, outside of the cell, there's a lot of these ions. So they're going to be diffusing into the cell. Versus this case where these ions are highly concentrated inside the cell, and so they'll start diffusing outside of the cell. So relative concentration will affect how fast they move across the membrane and what direction they go. There's also a voltage frequently associated with these membranes, right? Especially when we're moving charged particles, you can create kind of a voltage across these membranes. We're not going to get super into membrane potentials, but that's what that's called, right? You have these membrane potentials that are kind of created by the different charges of things passing through them, and that can change how fast things go in and out of the cell. And lastly, some of these ion channels can close. And so that's kind of a no-brainer. If you have a channel that can close and it's closed, then your diffusion rate is 0. If it's open, then your diffusion rate is going to depend on the concentration of ions and the voltage of the membrane. All right, so we've got these channel proteins, right? Tubes that can just allow ions and polar molecules to diffuse through and into the cell. All right. Conversely, we've got carrier proteins. Now, carrier proteins work a little differently. They bind selectively to molecules. So you've got a protein. Again, it's an integral protein, goes across the membrane. And it's going to be shaped to accept some specific molecule, right? And so the protein usually has some kind of like what we call an initial conformation. If we say conformation, when we're talking about proteins, we just mean the shape. Whatever shape the protein is its conformation. It is conformed to some shape, right? So they usually start in a conformation that is either open to the inside or the outside. So in this case, this protein is open to the outside of the cell. And some molecule that it is shaped to accept, right, binds in here. And so when that molecule binds to this protein, it changes the conformation, just the shape, right? So there's a reaction that happens here that causes the protein to change shape. So basically it just kind of goes open to the outside, then it opens to the inside and releases that molecule inside the cell. And then it's kind of back to the beginning, right? So molecule lands outside it, releases it inside. Or the other way, depending on the protein, right? Now, the diffusion rate, right? The rate of facilitated diffusion for a carrier protein will behave a lot like a channel protein up to a point, right? The more ions or molecules there are to move, the faster it'll go to a point, right? Because this isn't just like an open tunnel that things are just kind of like floating through, right? This is more like a stadium turnstile, right? Where like if you're going into, if you're like going to a game or something and you go through the turnstile, right? If there's just a few people, everybody just kind of walks through the turnstile and it's just as fast as people are walking through. But there will be a point, right, where the line starts to form, right? Because the turnstile actually kind of only turns so fast, right? So at high concentrations, there's kind of a limit on how fast things can be let into the cell because you've got this kind of turnstile movement happening with these proteins, right? So that's carrier proteins. All right. Now, even though channel proteins are open, they're still a bit selective, right? So whether it's a channel protein or a carrier protein, they tend to be designed for specific kinds of molecules, right? And so this again plays back to that idea that these membranes are selectively permeable, right? So they're selectively permeable in the sense that only nonpolar things can just kind of naturally diffuse through them, but they're also selectively permeable in the sense that we've got proteins that will bring polar things in, but only specific polar molecules or charged molecules, right? You don't want just any old thing coming into a cell. You just want the things that you need for the cell to function. So the membrane is selectively permeable frequently because these proteins that are bringing things into the cell are very specific to certain molecules. Let's see. Let's do a little osmosis. So related to diffusion, right? Because when we talk about diffusion, you're kind of talking about like the stuff in the water moving down its concentration gradient. You can kind of turn it around and talk about it from the other side, right? You can talk about what the water molecules are doing, right? Because they're involved in this process too, and that's osmosis, right? So your cytoplasm, right, the stuff in your cells, contains ions and molecules dissolved in water. Right? That's your cytoplasm. So it is functionally, kind of in the chemistry class sort of sense, an aqueous solution, right? The water is the solvent, and all the stuff in your cytoplasm are the solutes, right? Solutes are the things that are dissolved in water. And so osmosis is the diffusion of water across a membrane, right? So you can have water moving across a semi-permeable membrane, and when water has a membrane to move across, it is going to go to where there are more solutes. It's going to go to the higher solute concentration, right? So you can kind of picture it in your head, right? It's the opposite of diffusion. So if I have diffusion and I've got molecules that can cross a barrier, they're going to go from their high concentration to their low concentration, right? Until kind of water and solutes are balanced. If instead you have a membrane that will let water move, but not the solutes, right? So there's lots of sodium over here and not much over here. This water is going to move across the membrane until you have kind of an equal concentration of water to salt to sodium on both sides. Right? Right. So that is osmosis, where water will diffuse across a membrane towards higher solute concentration. Right, so let's illustrate that really quick. Right, so say you have a container, right, with a semi-permeable membrane. This thing will let water move, but not the things in the water, right? And so I've got a little sugar on this side and a lot of sugar on this side. What the water wants is to be evenly distributed around all of the molecules. So the water will move across the membrane, right? And this movement is osmosis. It will move across the membrane until it's evenly distributed around all of the molecules, regardless, right, of kind of where the membrane is. So if you have something set up like this, what you'll actually see, like in this kind of a container, is the water will be higher on one side than the other because it moves to where the solutes are. In your cells, this is facilitated by a special kind of little integral protein, an aquaporin, right? Which makes sense, right? Aqua is water, pore. So it's a protein created pore for allowing water, right? It's a specialized channel for letting water into or out of the cell. Aquaporins. We have a few minutes left, but I'd rather wait and start this on Wednesday. We can get all the way through it. So I'm actually going to stop there. And we will pick that up on Wednesday.